Hello and welcome to BIOS Frontier Science. We're thrilled to welcome Nora Disis, Helen B. Slonaker Endowed Professor and Director of the UW Institute of Translational Health Science and the UW Medicine Center Vaccine Institute to the show today. Nora, thank you once again for joining us.
Thanks for having me.
Of course, to kick things off, Nora, could you share a brief intro with us?
I'm Nora Disis. I'm a professor of medicine and an adjunct professor of obstetrics and gynecology and pathology, and I'm a member of the Fred Hutch Cancer Center here in Seattle, Washington. I've worked in the field of tumor immunology for the last 25 years or so, and in immunology in general for about the last 30 years. And it's been a wild ride, I can tell you, in terms of everything that's been going on in cancer immunology. So best career ever.
I love the enthusiasm. I'm so excited to dive in more and hear about the passion through your work. I mean, we're excited to have you, Nora, today. But before we dive too deep into specific projects, one question we really like to ask our guests, just throughout your career, what has been your North Star, really the common thread, if you will, that ties your work together?
I think without a doubt, my North Star has been that whatever I do has to impact patients in my lifetime. I started as a chemist. I was a chemistry major in college, did a lot of research in chemistry. I learned that I loved research, but I wanted it to have a more immediate impact. So today in the Institute, it's a big institute, it's got a lot of people in it. We're studying all aspects of tumor immunology. You know how research goes. There are so many different paths you can take as you tackle a problem. The one thing we always say to each other is, how will this go to the clinic, or how will this impact patients? And if you can't think of the answer, then put that in the garage and think about it a little bit more. And move forward with something else. So patients are definitely our North Star.
It's fantastic, Nora. Could not agree with the sentiment more. Want to dive into specific projects here and kick off this episode. So, going to pass it over to my co-host here, Reha, to talk about our first topic surrounding cancer vaccines in your research.
Hi, Nora. Thank you so much for coming and talking to us today. So, talking about cancer vaccines and, how that delivers, outcomes for patients, we wanted to first talk about HER2. So you were one of the investigators who originally discovered HER2's role in stimulating the immune system in breast cancer. And there are now several clinical studies focused on HER2 vaccines and T-cell therapies, and it's become an important drug target for immuno-oncology. So would you mind providing a bit of context for our audience on HER2's clinical significance as a target for immune systems?
Sure. So one of the things you'd like to do when you develop any type of target, whether it's a druggable target or an immune-associated target that you're trying to eradicate with the immune system, is to try to identify targets that have biologic significance to the disease. One of the things that we know is that in general, there are two types of antigens or immunogenic proteins in cancer. Mutated antigens, which can be seen as foreign to the immune system, or non-mutated proteins. The vast majority of the cancers I deal with, like breast cancer, ovarian cancer, prostate cancer, colon cancer, they are not highly mutated cancers. So the immunogenic proteins that the patients recognize are aberrantly expressed self proteins. We know that one of the major drivers to make a self protein immunogenic is if it becomes highly overexpressed. That means that epitopes that are available for T cell recognition that don't get into MHC molecules in high concentrations when the protein is expressed at basal levels, now do get into MHC molecules when the protein is really dramatically overexpressed. And we've published data that you can see immunity increasing in patients depending just on their baseline level of HER2 new protein expression in their tumors. So from an immune standpoint, it was a very good target because patients already had a pre-existing immune response to it. That's a benefit. It's also a detriment. But from a biologic standpoint, HER2/neu is a driver in about 30% of breast cancers. It's probably one of the major reasons why that breast cancer exists. It's a dominant oncogene that, when upregulated, really enhances signaling through the HER2 tyrosine kinase receptor and immortalizes that transformed breast cancer cell. So if you could create a modality, any modality, that could eliminate all tumor cells that overexpressed HER2, you would go a long way in being able to either eliminate that breast cancer or dramatically change the biology of that breast cancer.
Got it. And given that you said HER2 really is a driver for breast cancer, it has become a pretty important biomarker and therapeutic target, as you mentioned. So now we see, a number of clinical trials for HER2, and Translational, a monoclonal antibody targeting that gene, was approved in 2018. So I was wondering, how does the success of HER2 inform your thoughts about developing other molecular targets for use in immune therapy?
Sure. One of the things that trastuzumab and pertuzumab taught us is that they themselves act as vaccines. There are many publications that demonstrate patients who've been treated with Herceptin and nothing else can develop new levels of type 1 T cells directed against their tumors. And in fact, investigators at the Moffitt Cancer Center, Brian Sersnicki and his group, have shown that the greater the magnitude of that type 1 T cell response you generate with trastuzumab, the more likely you're going to have a longer disease-free survival or obtain a pathologic complete response. Now, we've studied vaccination to HER2/neu concurrent with trastuzumab therapy, and unfortunately, only about 40% of breast cancer patients get vaccinated by trastuzumab. So the addition of a vaccine to trastuzumab can either boost the immune response generated in that 40% or convert almost 90% of patients to HER2 responders from an immunologic standpoint, type 1 T cell response. So what that's taught us overall is, number one, overexpressed driver oncogenic proteins are excellent candidates for vaccines. And we have several multi-antigen vaccines now being designed targeting those overexpressed drivers. But the second thing it's taught us is that it's possible to integrate immunization into standard of care therapies. And I think we've come a long way from just looking at vaccines as, okay, now get a vaccine as a single agent. I think vaccines will be much more effective when they're integrated into standard of care therapies, because clearly many therapies augment and synergize with vaccines to even further boost the immune response compared to vaccination alone.
And I wanted to dive a little bit deeper into the point about vaccines, given you are the current director of the Cancer Vaccine Institute at UW. So recently, there are results from a pretty promising phase 1 trial for a cancer vaccine that you developed for HER2, and that suggested that your vaccine was able to generate a pretty strong response against HER2. I was just wondering, what really got you interested in developing a vaccine against HER2 in comparison to pursuing other modalities?
Well, science is serendipity, so my story is no different. When I started in the field of tumor immunology 25 years ago, the big question was, does the immune system play any role in cancer? And at the time, that was very unknown. In fact, it's amazing to think that in my career, we've gone to, does the immune system play a role, to now immune therapies are the backbone of most cancer treatment, right? That's in 25 years. So at the time, people were really intensely studying melanoma because there was very good evidence that patients generated endogenous immunity to melanoma, and several groups were beginning to identify in a very specific molecular fashion tumor antigens. And whereas tumor antigens had been identified previously in mouse models, in the mid to late 1990s, this was really the first time we were looking at humans and actually being able to molecularly dissect an immune response. And I was a student at the time, and I wanted in on it, right? And my previous training in graduate school wasn't in mouse models, it was in human immunity, humoral immunity. So I thought I could use humoral immunity as a probe to see whether patients developed immune responses to their cancers. And I was the only female in my oncology fellowship class, and I got put into the breast cancer clinic. So this was where I could get my material, which was blood from the patients. So I'd walk around with a bunch of blood draw tubes in my pockets, and I had a consent form for drawing blood to evaluate immunity to malignancy. And when we got done with the patients, my preceptor would let me stay in the room and see if I could get a tube of blood from them. So that really started my work in breast cancer. And what I basically did was to take a series of available human breast cancer cell lines and chop them up, lyse them, and run them out on SDS page Western gels at a variety of different resolutions, and just immunoblot to see whether I could get specific proteins. And one of the proteins that came up was this newly discovered protein, HER2. And many of the patients had very definite antibody immunity directed against HER2. From that point, we were able to develop capture ELISAs using a cell line called SKBR3, which just made high levels of HER2, and we found a human polyclonal antibody that we could use to create ELISAs to do validation. And then the hard work started of trying to raise T cells directed against HER2. And we were able to, they were rare, but we could generate HER2/neu specific T cells. So this documented that HER2/neu was a tumor antigen, but it also taught us the levels of immunity to HER2 from a T cell standpoint, especially cytotoxic T cells in patients, was very, very low. So the next obvious question would be, does immunity to HER2 have anything to do with controlling that patient's cancer? And my thought was, before we could really answer that question, we'd have to be able to boost immunity to at least measurable levels to be able to see whether immunity could control cancer. And just that question started my career in terms of cancer vaccine development.
Wow, that story is amazing, and it's really a testament to your determination in this field. So I totally applaud you for that. I just want to press on this point further a bit and, talk about all types of cancers, even though a lot of your work was in breast cancers. I was just wondering, how do you envision cancer vaccines fitting within the current treatment options for cancers?
I think that cancer vaccines can be used in 3 different areas. I personally have never really used cancer vaccines to treat an existing cancer. And there's a lot of controversy in my field. There are many investigators who think that a vaccine will only eventually be effective if you can take that vaccine and immunize someone who's got an existing cancer that's growing and have an anti-tumor response. As someone who reads all about all types of vaccines, that kind of didn't make sense to me, because I kind of liken it to if I went into an emergency room with an overwhelming pneumococcal pneumonia and the emergency room doc came at me with the pneumovax, I would probably call my attorney and say, this guy needs to be prosecuted here. I need the antibiotics. And in cancer, the chemotherapy is the antibiotic. So I'm not one who believes that you can use a cancer vaccine to treat an existing tumor. That was until immune checkpoint inhibitors came along. And of course, these drugs ligate different checkpoints. They're not only against, CTLA4 or PD1 or PDL1. There are many immune checkpoints that monoclonal antibodies are being made against to allow the pre-existing immune response to flourish. But getting back to my previous story, in many patients with solid tumors, unlike melanoma or kidney cancer, they don't have a really rich pre-existing immune response, or their pre-existing immune response is the exactly wrong phenotype that you want to expand. It's a type 2 immune response or a regulatory response. So before you could potentially get immune checkpoint inhibitors to work therapeutically in that population, those populations, you really have to give them or generate the type of immune response that you would like to be expanded. And there's really only two ways to do that. Generate that immune response outside the body and infuse it into the body and then try to expand it. That can be a little tricky in terms of toxicity, or you can create a vaccine that will generate a type 1 T cell response and then give it to them to expand. So for the first time in my life, after 30 years, I am investigating therapeutic vaccines in terms of the vaccines that we've developed and engineered to generate that type 1 T cell response, to team up with immune checkpoint inhibitors or other therapies to treat existing disease. But my wheelhouse has always been in secondary prevention. With the diseases I treat, such as ovarian cancer, breast cancer, the major problem with those diseases is disease recurrence. So they respond very well to standard therapy, chemotherapy, surgery, radiation. But in many cases, the stage of disease really makes the risk of recurrence high. So in the study we recently published in JAMA Oncology, where we immunized 66 patients with advanced stage HER2 positive breast cancer, stage 3 and stage 4, once they had completed treatment and either had stable bone-only disease or a complete remission, we vaccinated them. We were able to generate immunity in the majority of the patients, no matter what the dose. There were differences in the quality of the immune response depending on the dose. But at the end of the day, after an almost 10-year follow-up, over 75% of the patients were still alive. Over 40% of them had not recurred their disease. And in the most immunogenic dose, 85% of the patients were still alive. So we didn't even reach median overall survival at almost 10 years of follow-up, or median progression-free survival. So I think this shows that really targeting that prevention of disease recurrence is a sweet spot for cancer vaccines. And you certainly see that in terms of the new data coming out of some of Moderna's mutated antigen vaccines in melanoma, where they've also reduced the risk of recurrence by 40%. It was kind of interesting because the data were very similar to what we were generating with HER2. And that data is going to be presented and published within the next few months. So I think this is a very exciting time in terms of cancer vaccines for secondary prevention. But where our lab has moved in the last 6 to 8 years, once we've worked out vaccine technologies that can generate that right type of immune response, is identifying populations where we might be able to vaccinate to prevent cancer from even getting a foothold. And so now we have several vaccines that are really targeted for primary prevention. And these aren't viral antigens because, again, the majority of common tumors aren't caused by a virus. But these are really targeting biological drivers that have been defined in pre-malignant lesions, and either immunizing patients at high risk for the development of pre-malignant lesions, or immunizing patients with the pre-malignant lesions to prevent those lesions from progressing to invasive cancer.
Wow, that's really amazing how cancer vaccines can kind of span such a broad, I guess, types of therapeutics. I was wondering, for, I guess, translating cancer vaccines, both as a therapeutic or as a way to prevent further recurrence, or even as a preventative therapeutic. From your vantage point as a pioneer in the cancer vaccine space and as the head of translational efforts in the space at UW, where do you see the biggest challenges are in bringing cancer vaccines to market?
I think the biggest challenges are twofold. The first challenge is when you don't have a standard endpoint of tumor shrinkage, you lessen the enthusiasm for companies to look at a longer endpoint. So there are not very many companies that are going to stay in the game to go to a 10-year endpoint, right? And if you think about vaccines, the immune system in general, we've learned it does better with more minimal disease. So all the classic oncology endpoints, response rate, overall response rate, these types of things are not really suited to cancer vaccines. So we see a lot of these clinical trials now taking very high-risk patients. So these are definitely patients with disease that still remains that we can't measure, and going to a 2-year or a 3-year overall survival endpoint or a progression-free survival endpoint. And for industry, that's a long endpoint. And it also drives up the number of patients. So you're talking about randomized phase 2 studies with 200 patients to be able to get a signal to go on to a phase 3 study. So I think to really make vaccines a success, we have to encourage industry to be in it for the long term. The second major problem, I think, is in terms of preventative vaccines. A vaccine for cancer prevention in high-risk patients is even more of a long-term effect. Look at the tamoxifen studies to see if you could prevent secondary malignancy. They enrolled thousands and lasted years, and were done in the context of governmental funding, not necessarily funding by a company. No one could afford that type of study. And in our environment today, those types of investments are not so much being made in cancer research, these big mega studies. So we really need to do some work to find some surrogate biomarkers that will allow us to develop vaccines for prevention hitting a mark. So the studies can be smaller, they can be done more rapidly, and we can bring the best candidates to those bigger, more expensive studies. And I think there's a lot to be learned by the incredible data mining that's been going on, looking at anti-cancer responses with immune checkpoint inhibitors in terms of finding those potential biomarkers of response. But I think a lot more of this thinking has to go into primary prevention, where cancer vaccines, they might work the best. So those are the things now that I think we're focused on. The technology, the best ways to deliver vaccines, excellent adjuvants, how to engineer antigens to make them more immunogenic. The discovery now that's getting easier and easier of those mutated antigens that are going to bind to the appropriate HLA molecules. The technology has driven us to the point now where the biggest issues really are in the clinic.
That makes a lot of sense, Nora. I want to transition to our next topic, actually, diving further into your work surrounding T cell therapies and the tumor microenvironment. So the CVI is also focused on studying and developing other types of T cell therapies for oncological indications as well. And I just wanted to talk a little bit more about one particular approach, which has been to use T cells from a patient after they've been given a cancer vaccine to sort of prime the antigen T cells first. So do you want to talk a little bit more about what led you to this approach and also how priming the T cells actually impacts them?
Yeah, so what led us to the approach was in our very earliest vaccine studies, where we were using very old-fashioned technology, we had quite a few patients that once they were vaccinated did extremely well. They recurred their disease, but their disease grew much, much slower. And they always kept in contact with us and they asked for booster shots. They really wanted booster shots. And it was very difficult to be able to get funding from the National Cancer Institute on creating booster shots for different vaccine trials that we had run. So one of the things that we noticed is when the patients would contact us, we would ask them if we could take a look in their blood. This was now maybe 4 or 5 years after they were vaccinated to see if they still had an immune response. And we did that by purifying out their T cells and stimulating them in vitro with antigen and seeing if we could generate a T cell line. Now, in our previous work, and I kind of alluded this before, trying to generate T cells from patients, and this is prior to the era of trastuzumab being approved, was very difficult to do. I mean, you really had to take them through multiple in vitro stimulations. But from these vaccinated patients, we could take T cells and expand them up in one in vitro stimulation and have, pretty significant precursor frequencies derived from their peripheral blood. It was very easy to expand them. And then we just looked directly and we could still tell that they had measurable levels of immunity. So we thought that this would be a great way to be able to respond to the patients, to write a protocol to be able to take their blood. They had been previously vaccinated, expanded ex vivo, and we studied a variety of cytokine mixes to specifically stimulate type 1 CD4 T cells. Now, why did we stimulate CD4 T cells instead of CD8 T cells? CD8 T cells are the ones who actually are capable of killing the tumor. Well, in our early clinical trials, we also found that even though we were immunizing with a vaccine that was specific for HER2/neu, when we developed the technology to look at immunity to multiple antigens, we found that the patients developed new immunity after HER2/neu vaccination to multiple different antigens that were potentially expressed in their tumor. And this is a phenomenon that's called epitope spreading. And it's the basis of how you get autoimmune disease. That you have a driver type 1 CD4 T cell that's capable of trafficking to tissue. It's secreting cytokines like interferon gamma and IL-12 and TNF alpha, and you're activating antigen presenting cells in the tumor microenvironment and converting them to professional antigen presenting cells, and they begin presenting all the antigen available in the tumor. So we thought, all we have is HER2. Let's get really juiced up type 1 T cells, expand them up to great numbers, and hopefully will induce massive amounts of epitope spreading so that they'll have high levels of T cells of all specificity being able to attack this tumor, which was existing and no longer responding to treatment. And the initial results were fascinating. We had patients where we could measure the level of T cells in their blood after 3 infusions for over a year, and we had patients who responded. And we went ahead and we did a phase 2 study of the approach in 26 patients which just recently got accepted to clinical cancer research. And the results, again, are quite interesting, with just a significant stabilization of disease and some responses. So that is a viable. The reason why we chose that rather than CAR T cell therapy directed against HER2 is that these cells are not high avidity T cells. So the chances for off-target toxicity, which are a major problem with CAR T cells, simply aren't there when you use autologous T cells. The problem with autologous T cells is that you just can't get enough of the right type of T cells for expansion. So doing this concept of vaccinating first to generate that T cell population and then come up with the magic sauce ex vivo to expand them seems to really have an effect on the clinical outcome of the patients.
It's really amazing to hear the storied history of your implementation of T cell therapies into your research groups via the CVI. And it's super exciting to hear your perspective, especially. So, with that history in mind, we'd love to focus into your current initiatives you're approaching with immune modulation strategies that can make a tumor appear more dangerous to the immune system. So, could you also elaborate a bit on what types of strategies your group is exploring?
Sure. For a long time, we've had a big interest in different types of adjuvants. The adjuvants that we use for our vaccines are GM-CSF because we give the vaccine intradermally, and that acts as a big chemoattractant for dermal dendritic cells and skin Langerhans cells. And given intradermally, GM-CSF has been a very effective adjuvant in contrast to giving it subcutaneously or intramuscularly. We also study adjuvants such as IL-12, things that will specifically activate type 1 dendritic cells, because our vaccine ploy, as I talked about, is really the stimulation of CD4 T cells to generate that epitope spreading, which we've shown is associated with longer survival if you develop epitope spreading. We also have done a lot of work looking at toll-like receptor agonists, and this drew us to developing DNA, nucleic acid-based vaccines, as our mode of delivery rather than chemically synthesized peptides or proteins, because the DNA itself has adjuvant properties in terms of toll-like receptor stimulation. And we still continue to work on looking at different toll-like receptors in combination in terms of adjuvanting vaccines. But I think our biggest breakthrough recently has not been in identifying stimulators, but actually in identifying problems. And one of the things that advanced our vaccine research was the understanding that within the natural sequence of these overexpressed self proteins, I mean, they're self, so they're not like mutated antigen vaccines. They're not seen as foreign. But one of the reasons why they're not seen as foreign is within the natural sequence are epitopes, CD4 T cell epitopes, that stimulate regulatory responses. T cells that induce high levels of IL-10 and IL-6, and these antigen-specific T cells are highly avid for the MHC. So if it's a competition between an interferon gamma-inducing epitope and an IL-10-inducing epitope, the IL-10-inducing epitope is going to win. So what we discovered was these self-antigen vaccines must have these regulatory epitopes edited. If you don't, you'll never get high immune responses. But if you can identify what we call Th1 selective class 2 epitopes, these are epitopes that only induce type 1 T cell responses, you can end up with a vaccine that generates high levels of type 1 antigen-specific immunity, as high as you would see to a mutated antigen vaccine, without any induction of IL-10 or any antibody formation, which is kind of a surrogate of the generation of a type 2 immune response. Once we made that discovery, we went into vaccine production mode. And we have taken now about 50 antigens, and we've identified their Th2 selective epitopes, we call them, their Th1 selective epitopes that are going into cancer vaccines. And it gave us a database to allow us to start addressing what makes a Th1 selective epitope and what makes a Th2 selective epitope. And what we found was that many of the epitopes that induce IL-10 actually have a high degree of homology to bacteria in our microbiome. Now, we know that we need T cells in our microbiome to prevent the development of immunity to bacteria, right? Or we'd all have colitis and we wouldn't have functioning guts and we'd be in a lot of trouble. But we also know that these T cells can become peripheralized. What we didn't realize is many of those T cells share a high degree of sequence homology with common tumor antigens. So you can imagine you have your gut, you have peripheral tolerance in the gut, you have lots of antigen-specific T cells directed against bacteria, preventing an inflammatory immune response against these bacteria. But there are sequences in those bacteria that are highly homologous to antigens like SOX2 and YB1 that become markedly upregulated in tumors, even at a very early stage. So in essence, your gut immunizes you against your cancer in a bad way. So if you go to the TCGA and you look at the immune phenotype across 10,000 tumors, and this was done by Dr. Vestin Thorson and colleagues in the Immune Landscape of Cancer project, you'll find that the majority of tumors, even at the earliest stages, are infiltrated with type 2 T cells, not type 1. We think the reason why that is that as soon as those proteins began to overexpress in the tumor, the T cells in the gut that had already been primed to those sequences began to traffic to other areas of antigens like a good T cell would do. But unfortunately, secreting cytokines like IL-10, they prevent the development of a tissue-destructive immune response and basically cause tumors to grow. So we've spent a tremendous amount of time creating blood tests to be able to measure these T cells in the peripheral blood of cancer patients to see if those T cells are at a level where they could cause a problem, and in many patients they are, and precision probiotics directed using bacteria where we never see these homologies to decrease this memory T cell population and be able to allow immune responses to develop in the tumor that may be more productive by decreasing the level of these, we call them bacterial tumor antigen cross-reactive T cells. And the early results of these probiotics are very exciting. And so we hope to have these in clinic for every cancer patient where we measure a high level of these T cells to be able to decrease those T cells over a period of weeks so they'll respond better to any therapy, not only immune therapy, but chemotherapy, where we know having a functional immune system is extremely important.
It's absolutely impressive how many different avenues your group has been exploring. Just kind of going off of that, given these different research areas and so many different stakeholders involved in translating these novel vaccines and T-cell therapies, thinking more broadly, how do you approach bringing in and working with different PIs within the Institute as the director of the CVI?
Well, I think that one of the benefits of the Cancer Vaccine Institute is that everyone has a say. It doesn't matter if you're a PI, doesn't matter if you're a junior research scientist. We bring people in to the CVI that add something. Most of the PIs who work in the group, we work this as a big group that's based on big goals. And each of the PIs has their own individual interest, but we also work with that North Star idea of getting things into the patient. So if you look at someone like me, I'm more attracted to the lab. I don't have the energy, time, or the know-how to design some of these really complex clinical trials that we need to answer questions. So we bring in people where that's their expertise. And I think if you create a lab environment full of very diverse people, toxicologists, pharmacologists, regulatory specialists, immunologists, people who do biomarker development, you don't get into a hierarchy because everyone is an expert in their own right. And when I have a question, I go to the expert. I mean, I don't try to reinvent the wheel. I don't try to do it myself. And what happens is you end up with a very integrated, interactive environment where people depend on each other and everyone feels like they're contributing significantly to a big goal. And we are goal oriented. We've got a project manager that for every single project, we've decided what the big hairy goal is at the end of that project. And we have project meetings where we talk about where we're slowing down or where we're speeding up and how we can use, let's say, philanthropic dollars to speed up that process or troubleshooting issues early in terms of being able to figure out, is this the right avenue to take or the wrong avenue to take? Because I think we all feel an incredible urgency. You know, one of the common things you hear in the lab is we'd all love to be out of business. I mean, we've got an autoimmune program going on. We'll switch the whole thing to autoimmunity if we can crack this cancer nut. There's always something to do. But we're serious about curing cancer. And when you see the patients every single day, you know, the 32-year-old with 2 kids with stage 4 breast cancer, you go back and you're like, don't turn off the lights. We got to try to save her, you know. These are the people who really fuel what you do. And I think that urgency and immediacy, and especially the discoveries that have been made in the field, even in the last 5 years, it keeps us all up at night and makes the work go faster. And I think everyone feels like they're really doing something. And that's what a lab group should be.
Well, would love to, but yeah, it's amazing the type of culture you've been able to cultivate there. You know, just diving a little bit more into this talk of like speeding up translational research into the clinic and leadership. You're also the director of the UW Institute of Translational Health Sciences, or ITHS. So I was wondering, you know, how has your experience as a clinical investigator and the founder of a company translating your own research shaped how you approach leading the ITHS?
It really did shape how I led the ITHS, because when I started leading the ITHS, which is part of the CTSA program from the NCATS, the National Center for Accelerating Translational Science, most of the resources around clinical and translational research were either non-existent or very disparate within the academic medical center. You know, people developed resources and they used them, and they wouldn't mind if other people used them, but no one knew about them. And the path forward for translational research wasn't clear. The very first time I wrote an investigational new drug application, I called a very senior person who I'd met at a meeting and asked her if she'd ever put one in. And she kindly shared with me all what the steps were, what you had to do, and it was really like being in the dark. So I feel that centers like the ITHS, which are at 60 academic medical centers around the United States, really are a source where investigators can go and say, how do I take my discovery from the bench to the bedside? What are the steps? Who's available to help me? And how do I do it? And I'm proud to say that I think the Institute for Translational Science at the University of Washington has all the components in place to be able to have investigators come, and those especially with that drive to try to figure out how to make their research more relevant to the patient, at least have a starting point to be able to do that. And I also think the University of Washington is very friendly to people who are entrepreneurs. And let's face it, there is no academic place in the world that can manufacture a drug and market it. So at some point, there's got to be a handoff to someone who's interested in doing that. That's a whole other type of science. And I know a lot of academic institutions, dealings with industry is kind of a dirty word. And I think that has to go away. A lot of discovery comes out of the academic medical center. There's a lot of discovery here that's sitting on a plate, getting stale, that could really make a difference. And I think the infrastructure of the academic medical center has to do more in finding those partners who are interested in that idea and can nurture that idea and take that idea to its fruition. I mean, if you look at immunology, immune checkpoint inhibitors, those came from academic labs. You know, some of the mutated antigen vaccines that we're talking about now, academic labs. So industry is open and willing. We just have to make the pathway forward easier to be able to do this type of final translation to product.
And I wanted to think a little bit more about how you mentioned that, academic labs should be able to partner with, different industry as well as, other institutions. And it seems like the ITHS is the center at a lot of different collaborations amongst different institutions and communities. So I was wondering, how do you manage interfacing with all these stakeholders in order to accelerate translational research?
Well, the CVI has about 40 people working in it. The ITHS has about 200. So it really is a different scale. And we do break up our programs very specifically with leaders and collaborators in those programs. As important as industry taking a product forward is the community willing to accept a product. So one of the things that we often overlook, you develop something and you say, oh, I'm going to run a clinical trial. And of course, everyone will want to be involved. But, if your product requires that you get T cells removed every other day and you've got to be hooked up to a machine and then they have to be generated and there's a 30% chance you're not going to get those cells, that's not something people are interested in. So I think community members or stakeholders who are the end users need to be integrated into the development process much earlier than what they are now. Right now, they're integrated in phase 1 clinical trials where the product is already almost fully baked, and then you go out looking for people who want to see if it works for them. I think that we need to bring in the community as full partners in clinical research. Much of the research that's being translated is being paid by their tax dollars. And it just frustrates me so much when I hear my neighbors like, research, why would we ever pay for research? Because it's really doing something for you. We need to communicate that better, and we need to bring the community into the research that they care about. But the second thing is, the more the community or stakeholders or end users of our potential products, like cancer patients, understand what we're doing and really see the potential of that discovery, the more they'll volunteer for clinical trials. And that's one thing that is a problem. I mean, it's a problem just starting a clinical trial with all the regulatory, but communicating to the patients the importance of being on a clinical trial. You know, physicians are in their office, they're seeing a patient every 15 minutes. A lot of times the patient has to be the one to come in with the list from clinicaltrials.gov saying, what about this? What about this? What about this? Many people don't even know that they can enroll on a clinical trial. And I think that that is an example of how little we as scientists are talking to the community, educating the community, and engaging the community. And I think that that has to change dramatically. And the
That
ITHS
makes
is trying to do it.
that makes a lot of sense. It definitely goes back to your original North Star about patient-centric approaches and you're developing for the patients. So I want to talk a little bit more about the clinical trials that are being run at the ITHS and how you approach that, as your express mission is to speed science to clinical practice. And also I wanted to talk a little bit about, given your experience as a founder of your own company, Epiphany, I wanted to understand what types of outstanding inefficiencies and challenges do you see generally that exist in clinical trials? And, potentially as a closing thought, do you see any room for technology to alleviate some of these issues?
So the first thing is what type of studies does the ITHS run? We don't actually run studies, we facilitate those studies. Many of these studies are taking place in our partner institutions, which are the University of Washington, the Fred Hutch Cancer Center, and Seattle Children's Hospital. But we're trying to push those studies farther into the community. And we're doing this by creating community research networks. These are people out in the community who are very interested in research. They're already doing research for companies, and especially for us in the Pacific Northwest, outside of the Seattle area, much of our reach across our 5-state region, which is Washington, Wyoming, Alaska, Idaho, and Montana. It's called the WWAMI region. The University of Washington is one of the only medical centers and medical schools for this region. We are connected with them, and these regions are very rural. And many of these regions and their major hospitals have started small clinical research centers to bring in new drugs and for companies to be able to test whether this would work in a less resourced or intensive setting. So we've started trying to move academic clinical trials out to the region. Many of these can be life-saving. So for instance, during the COVID pandemic, because we already had these existing clinical research center networks, we were able to get some of these life-saving drugs in terms of clinical trials rapidly established at some of these more rural clinical research sites and available for people who were dying of the COVID infection. So I think we have to work locally, but think much more globally in terms of how we can conduct clinical trials, where we can conduct clinical trials, and most importantly, simplify our clinical trials. And I think we can do that. Will technology help make things easier? Of course, it already is. Clinical trial management systems, having outstanding CROs, or now these new technologies that will allow us to consent patients or follow patients through teleresearch. These are very, very important endeavors that should be able to expand our reach. And certainly a small startup company like Epiphany, many of these things just are not something that a company with limited resources can get their hands on. So for us, it's been very critical to continue our academic work and be able to have Epiphany front face that work to the pharmaceutical companies. And with that, make those key collaborations that will take our academic work to a product. And I think out of academia, spinning these type of startups that are that front face that can go out while, I'm in the lab continuing to do what I do, and really spend the time looking for the right partner has been just transformational in terms of taking many of our vaccines to randomized clinical trials.
It's amazing to hear from your perspective, Nora. I mean, thank you again so much. I genuinely wish we had more time to dive into the amazing work, and I really could not stress even more the mantra, the guiding principles around translation and getting into patients is just so inspiring hearing from your own words. So just really to close things off here, we want to ask a few quick rapid fire questions, just to tie a little bow on this episode. But first, as you mentioned before, I know you had a quick shout out, a quick call to some other folks in the industry. As we're talking about quick ideas around translation here and some ideas kind of sitting on the shelf, do you have advice for industry professionals seeking to reach out to help assist in your work, or any other calls to industry or individuals? Just want to give you that platform.
There's lots and lots of conferences that are bringing industry and academia together. And I think academics have to reach out to people they think would be a good fit. And I think industry also needs to broaden its interest and really try to do a deep dive into different types of technologies that they're interested in. So I think there are methods out there to be able to create these connections, and it's easier than ever before just to directly contact people. So people contact me all the time, and I'm delighted to talk to them.
Well, it's amazing to hear, Nora. And just while we're on that vein about reaching out, I mean, any other closing thoughts or shameless plugs? How can our audience hear more about your work?
Well, you can always go to our website, which is www.uwcbi.org. We always list our open clinical trials on that website. Everyone's phone number in the lab is listed on that website. And we love talking about what we do. And if anyone's ever in Seattle, come by for a tour. We love to give lab tours and give you a good cup of that Seattle coffee that we all live on.
Oh, of course. Oh, it's famous. Well, I mean, just thank you again so much, Nora, for an absolutely fantastic episode. I'm sure our listeners enjoy this as much as we did, but thank you again. Just grateful for your time.